All right, class, welcome to Chapter 7, where we're going to talk about the structure of the biomembrane, kind of moving past a lot of the stuff we did before. That was a little bit of review of genetics and a little biochemistry, etc., and get into some new content here. Okay, so let's look at kind of an overall view of the biomembranes. So a plasma membrane it defines the cell and separates the cytosol from the external environment. So here we have the external environment, and on the bottom here is the intercellular space in the cytosol. Biomembranes have the same basic architecture as the plasma membrane. So these are membranes of organelles and things of that nature, which is a phospholipid bilayer that's about 3 nanometers thick, and it has embedded proteins in it. So this membrane is fluid, so things can move around within this membrane. It's not like a solid structure, and it's got a fluidity that's kind of similar to olive oil, if you've, you know, how viscous olive oil is. So there are non-covalent interactions between phospholipids and between membrane proteins, and this provides the membrane's integrity and resilience. And individual phospholipids can diffuse laterally within the plane of the membrane, so things can move within that plasma membrane, but they can't leave because the hydrophobic the hydrophobic tails want to stay together, right? So they can move within the plane, but they can't go up or down, so two-dimensional movement. The hydrophobic core prevents this unassisted movement of water soluble substances from one side to the other. So anything that's soluble in water can't get through the membrane because that hydrophobic core, where all those tails are, won't let it. It's kind of pushing anything water soluble away, because I think water soluble is polar, right? And so the non-polar tails won't let anything pass through. So in terms of proteins, membrane proteins provide the cellular membrane its unique set of functions. Integral membrane proteins, such as transmembrane proteins, span the bilayer. So this would be an example of a, looks like an ion channel of some sort, that is spanning the lipid bilayer. And these often form dimers and higher ordered oligomers. So we have our example of a dimer transmembrane protein over there. There are also lipid-anchored proteins, and that is this green guy right in the center here. And this is a protein that is tethered to one of the leaflets, so we call each of those phospholipid single units, monomers, leaflet. So they're tethered to one leaflet by a covalently anchored hydrocarbon chain. And then, so those are kind of our surface proteins of the cell. We also have peripheral proteins. So we have two examples here. And these, there's one on the external environment and one in the cytosol. And these are associated primarily by specific non-covalent interactions with membrane proteins. So they are attached to membrane proteins as kind of a second layer, right? So they're not directly involved with the membrane, but they are involved with the protein that is involved with the membrane. And these can also tether things. So in this example here, we have the bottom cytosol example. We have a peripheral membrane protein that's tethering that, or connecting this dimer membrane protein, the red one up here, to the actin cytoskeleton within the cell. So eukaryotic cellular membranes are very dynamic structures. The membrane is very fluid and very flexible, and this enables organelles to assume their shapes and provide this dynamic property that enables membranes to do budding and fusion, so things for like endocytosis, which is important. So in our example here, we have an HIV-infected cell, and this cell the HIV is infecting the plasma membrane, and we have the virus core, the HIV core, that is enveloped by a region of this plasma membrane that contains within here specific viral proteins, and the HIV particles will bud off from the plasma membrane. And then in cell B here, we have a stacked membrane of the Golgi complex and budding vesicles that are involved in intracellular trafficking. I'm trying to find my mouse here. There. So those are the budding vesicles that are occurring, that are these membranes surrounding whatever proteins or products that the Golgi is trying to package and send different places. So it's a very moldable and dynamic structure of the plasma membrane. So let's take a closer look here at the bilayer structure of these biomembranes. And so amphipathic molecules are ends have different chemical properties. So with these phospholipid bilayers, there's the hydrophobic or water-fearing fatty acid based hydrocarbon tail, which are the two-pronged tails that are coming off the fatty acids. And those partition away from water because they're water-fearing, hydrophobic. And then there is the hydrophilic or water-loving polar head groups that interact with water molecules. And so we have a picture here of an erythrocyte membrane. Cool. And the stain used for this this photo here interacts with the hydrophobic heads, but not the with the hydrophilic heads, and not with the hydrophobic tails. And this yields this railroad track or two-lane appearance of the membrane between the exterior and the cytosol, because there's heads on both sides and then the tails go into the middle. So one track is one set of heads and the other track is the other set of heads. And so phospholipid structures, they form spontaneously, right? Because they're driven by this hydrophobic, hydrophilic dynamic. And so they're driven by the behavior of these hydrophobic and hydrophilic ends that are exposed to water. And their nonpolar tails will closely pack to stabilize. In the interactions that cause them to stabilize are the van der Waals and hydrophobic effects. And interactions between the hydrocarbon chain polar head groups. And for the interactions between the head groups are ionic and hydrogen bonds that are keeping those intact. So for the tails, it was van der Waals and hydrophobic effects. And for the heads, it is ionic and hydrogen bonds that force this structural organization. Exposed hydrocarbon fatty acyl chain, the edge is thermodynamically unstable. So this form here is not stable. This is just kind of a cut, right? So you have to have an entire round spherical structure for this to be stable. And so what happens is you can really have two different spontaneously forming structures from these. One is a micelle, and this is where it's only a single layer, so it's a phospholipid single layer, and this usually occurs when you have fatty acids that have shorter tail groups, hydrophobic groups. And so you see this when you're doing the dishes and you put dish soap in there, and there is you have like an oily pan and you see those oil bubbles. Those are micelles, and you don't see them a ton in biology because mostly you see phospholipid bilayers. And so we have an example here of a liposome, and that is where we have, similar to the plasma membrane, two tail groups going together, and the head groups on the outside. And so in this liposome example, we have an aqueous compartment in the center. And if you blow this up real big, you'd also have the phospholipid bilayer of a cell, right? And so this would be the extracellular compartment, and this would be the cytosol on the inside here. And so this is just a cutaway, and that is not thermodynamically stable. It will either form into a micelle or into a liposome. Okay, what if we want to study phospholipid bilayers? So one thing that we can do is we can take our cell and we can treat it with an organic solvent. And what that will do is it will break apart and solubilize the phospholipids and cholesterol, while the proteins and the carbohydrates, they fall out of solution, and then we can just throw those away. The organic solvent that we can use, an example would be a 3-to-1 mixture of chloroform and methanol. So they are hydrophobic or non-polar solvents that would dissolve this. And then we remove the solvent through evaporation. And once we do that, we've isolated only those phospholipids. And you can disperse them back into water and look and analyze how they form these liposomes and how they kind of reconstitute into this bilayer. Or what we can do is we can have this vessel set up here where we have kind of a beaker that has a plastic partition halfway through it, and within there, that plastic partition is a small hole, and these lipids are going to want to put their hydrophobic tails together really, really bad, right? They're going to want to either try to form liposomes or find any way that they can to put those hydrophobic tails together. So what we can do is we can look and see how they form these bilayers that would be present in a cellular membrane, because they will stick their tails together to cover up that hole in the plastic. And so you can see that kind of is a representation there of a section of phospholipid bilayer. So let's look at a few of these different types of cellular membranes. So we've talked a bit about the just the cell membrane here. As you can see, we have the cytosol that is this kind of tan color, and then we have the extracellular space, or the extracellular environment, the aqueous environment that is on the outside here. And there are other organelles, though, that also have these bilayers, right? And so there are some that are very similar to the cell membrane, such as the lysosome here, the Golgi apparatus here, where they have an internal aqueous space, and then they have a cytosolic face that is facing the cytosol. The same can be said for the endoplasmic reticulum. And however, there are other organelles, such as the nucleus, the mitochondria, and the chloroplast, which they're not showing here. They have two phospholipid bilayers. So they have a outside layer, and then they have this intercellular or intermembrane space, which is between the two bilayers. And as you can see down here on the nucleus and on the mitochondria up here, there is a space between the inner kind of cytosolic environment and the external cytosolic environment of the cell. And so there's really four phospholipid pieces, or two bilayers, between the inside of these organelles and the outside cytosolic environment. So when these membranes do have events like budding, so during endocytosis or exocytosis, the faces of the membrane are preserved, right? So in our example here, we have this membrane protein that has an exoplasmic segment and then a cytosolic segment. And so this cytosolic segment always wants to be facing the cytosol, and then this exoplasmic segment always wants to be facing water, this aqueous external environment. So when we have the process of endocytosis, where the membrane is invaginating and taking in something, say there is something, protein or something it wants to take in from the outside. As you can see, the orange side here, the orange portion of this bilayer, of the two-layered membrane, is facing the cytosol, and even through the formation of this, this vacuole, that orange portion remains in contact with the cytosol. And if we were to look at it from a protein perspective, to start here, we had the protein on the top right that had the cytosolic segment, the big round ball, facing the cytosol, and even through this process of endocytosis, that portion still faces the cytosol, and the exoplasmic segment, those two little balls, are still facing the aqueous solution. And the same can occur through exocytosis. So if this this vesicle were to go in the opposite direction, the exterior segment, or the exoplasmic face that is facing the aqueous solution, or the lumen on the inside, still faces that as it merges with the phospholipid bilayer, or the cell membrane. So while all these membranes are phospholipid bilayers, they still have a lot of variance in between them depending on cell type. So our examples here is we have an erythrocyte cell, and as you can see, it's a very smooth, flexible shape, which is required for squeezing through blood capillaries with very small diameters. And then we can compare that though to a cilium membrane. And so the cilium membrane here is from ependymal cells that are lining the brain vesicles, and they have these regions of plasma membrane that project from the cell these cilia that are enabling ciliary motility. And so as you can see, it's a lot rougher shape, and those cilia are these, you know, they have the motor proteins down at the bottom that are anchored in the cell, and then these would be structures that are ancillary or attached to those motor proteins. You can see it's a much rougher surface. So it's even though we look at it in all these diagrams and we see this bilayer and we think, okay, you know, this is the same for everything, they're very customizable and very different depending on cell type. Okay, so there are really three classes of membrane lipids in a typical biomembrane. So these are three classes of amphipathic lipids, which are phosphoglycerides, sphingolipids, and sterols. And these differ in the structure, abundance, and function within the biomembrane. So we'll start with the phosphoglycerides, which are the most abundant, and they form this glycerol backbone. The tails have two esterified hydrophobic fatty acyl chains. So those are the two chains that we see in all these diagrams that we've looked at. In these esterified hydrophobic fatty acyl chains, they vary in length, although they're commonly 16 or 18 carbons long. And they also vary in saturations. We've talked about saturated, which is no double bonds, and unsaturated, which has 1, 2, or 3 double bonds. Unsaturated being the good fats, and saturated fats are not as good health-wise. The more unsaturated it is, the more fluid it is at room temperature. They have 4 major head groups, so those change depending on the type of phosphoglycerol. And so we see we have PE, PC, PS, and PI. So PC is phosphatidylcholine, PE is phosphatidyl ethanolamine, and then PS is phosphatidylserine, and then PI is phosphatidyl inositol. Excuse me. We also have kind of the subgroup here, the plasmalogens. So the only difference between the phosphoglycerides in the plasmalogens is how the hydrophobic tails are attached. So here we have at the top, we had two ester linkages, and down here, the two chains, as you can see, which is different, is we're missing that double bonded oxygen. So one has an ether linkage and one has an ester linkage on the two tails. So those are just kind of slight variations or a subgroup of these phosphoglycerides, excuse me. So we also have sphingolipids, and these are derivatives of sphingosine, excuse me. And the difference between this and the phosphoglycerides is the attachment point again. And so here we have attachment by an amide group as opposed to by the ester or ether or ethyl linkage from the phosphoglycerides. And they also have differences in the head group. And so we have up here this SM, which is sphingomyelin, and this is just a different head group compared to the PS, PC, PE, PI of the phosphoglycerides. But also with the sphingolipids, we also can have sugar head groups. So here we have kind of a glycerine or glucose attached head group to the hydrophobic chain. And so just slightly different conformation compared to the phosphoglycerols, where we have a different attachment point and different head groups. And lastly, we have the sterols, which are membrane components. And in animals, we have cholesterol. In fungi, we have ergosterol. And then in plants, we have stigmasterol. So here we have cholesterol, ergosterol, and stigmasterol. And these sterols have this amphipathic structure where the head group is a single polar hydroxyl tail. Where's my marker? Here we go. And it's conjugated to a 4-ring hydrocarbon and short hydrocarbon chain. So you can see here, they all have the similar structure where they have this 4-ring carbohydrate or hydrocarbon group, but they also, what's different is you can see there are slight modifications in if they have double bonds, such as here. So these are much more unsaturated, while as you can see, cholesterol in animals has no double bonds, so it's it's more saturated. And the functional groups are kind of in different positions between the different kingdoms. The phospholipid bilayer has two different types of consistency. It's got this gel-like consistency and a fluid-like consistency. And so the lipid molecules within the membrane, they move around a lot. It's very fluid, and they exchange places with their neighbors about 107 times per second. So they're moving all over the place. And this lateral diffusion can result in movement of several micrometers a second. So they move quite a bit throughout their layer. And so these diffusion rates indicate the bilayer is about 100 times more viscous than water. So it's kind of like the viscosity of olive oil, like we had said earlier. However, while we do see these movements kind of back and forth between these the monomers of the phospholipid bilayer, we rarely if ever, see them diffuse across like this. And that's because there's this high energetic barrier to moving the hydrophilic head through that hydrophobic core. So rarely will you see a polar head group that was exposed to the external environment switch to the cytosolic environment. And so there is this gel to fluid transition. So if you have phospholipids with long saturated fatty acyl chains, they assemble into this highly ordered gel-like barrier here. And so this is kind of what we've seen in all of our diagrams before. But if you are to heat up this, this phospholipid bilayer, it causes the tails to become disordered, which we see on the right-hand side here. And it induces a transition from a gel to a fluid. And this happens within a temperature range of only a few degrees. So you go very quickly from being solid to hitting this temperature threshold and becoming very liquid. And when this happens, this fluidity or increased fluidity and disordering of the nonpolar chains, it causes the phospholipid bilayer to become thinner. So as you can see, kind of in this representation at the top here, but also in this kind of molecular structure or model down at the bottom, you see it's a lot thinner when those tails become disordered because they're becoming more kinked and not as straight to the point. The shorter the more wandering those tails become, the less distance they can travel. So one way we can study the fluidity of these membranes is with this FRAP, or fluorescent recovery after photobleaching experiment that we talked about previously in, I think, chapter 4, if I'm not mistaken. I don't remember exactly. But anyways, so the way this works is with our experimental protocol is we have our cell that we want to look at the fluidity of the membrane by studying these membrane proteins. Next, what we do is we label those membrane proteins with some sort of fluorescent reagent that's either linked to an antibody or GFP tagged, etc. Then what we do is we take a laser and we bleach a small portion of that cell. So we lose all fluorescence in that area, but the rest of the cell, the fluorescence remains intact for those proteins. And then we watch and see where we photobleached that spot, how long it takes for that fluorescence to recover, or at least partially recover, from other proteins moving in, because they're able to move throughout this membrane. So if we were to look at the results of an experiment like this, we'd see that at the very beginning of our experiment, which is akin to this phase here, we would measure the fluorescence, and we see we have this baseline for fluorescence of just under 3,000 ARB units. And then we do our bleaching, and then we, over time, measure that area to see how much of that fluorescence returns. And so in this case here, we see that 50% of our fluorescence returned after 3 minutes, but we never recovered 50% fluorescence. And so that may indicate that 50% of those proteins that we had stained are not mobile, and they may be anchored to the actin cytoskeleton within the cell and are unable to perform this fluid movement that non-tethered proteins are able to perform. So we've talked about how there are different components of the plasma membrane, but different cells have different combinations of these components. And so as you can see here, we have PC, PE, and PS again, and we also have cholesterol, and you can see that in human cells we have a lot of cholesterol. In plant cells we have no cholesterol. Oh, and I circled E. coli there too. But there's no cholesterol, and that's why eating plants is good for your heart, right? Because you don't get high cholesterol, especially in addition to, I should say, saturated fats. And if you are to look at rats, they have a little bit of cholesterol within their plasma membranes, but humans have quite a bit. And then you can see the different phosphoglycerides or phospholipids combinations kind of vary between different organisms here as well. So this is just kind of an idea to show you that different animals, different cells within those animals, all have different amounts of these components that make up the phospholipid bilayer in the plasma membranes. So lipid composition can affect the thickness and curvature of the bilayer. And so it differs in the exoplasmic and in the cytosolic leaflets. This influences the thickness and the membrane protein distribution as well. And so in our first example here, if we have pure sphingomyelin, I'm having some rough time with that word, sphingomyelin layer, we have on the right-hand side, you can see it's quite thick, and we have some cholesterol interacting with that. But if we compare that to PC, we have a much narrower membrane. However, with PC, if we add cholesterol, that increases the thickness of the membrane from 3.5 nanometers to 4 nanometers. So the different compositions here will increase or decrease the thickness of that membrane. So in terms of PC, they have this kind of cylindrical shape here, as you can see. And that it makes it so they form very flat monolayers. And if you compare that to PE, they form these cone shapes, right? So the tails are wider at their ends than they are at the polar heads. So they're that cone shape. And so when you put many of these PE together, they form this natural curve, as you can see here. And so if we were to look at this in terms of a bilayer, where there are turns or a lot of these cone shapes next to each other, we form turns where the inside are the PE and the outside are the cones. And as you can see, the way that this is kind of set up, that this satiates or this fulfills both PCs want to be inline cone, and PE's desire to be, or sorry, an inline cylinder, and PE's desire to be this cone shape while maintaining the integrity of that plasma membrane. So there are enzymes that cleave these phospholipids, and they are called phospholipases. And they cleave ester bonds that connect the acyl chains and the head groups. And the different phospholipases have different activities. And so each type cleaves a specific bond. And so you can see here that phospholipase D cleaves the bond between the phosphate and the oxygen, while C cleaves that same bond but further away from the polar head group. We have A2 that cleaves an oxygen carbon bond on the side chain, and then we have A1 that also cleaves an oxygen carbon bond but on the main chain and not on the side chain. So in cells that have excess lipids, we get the formation of lipid droplets. And these form by budding and scission from the ER membrane. And so cells store excess lipid in these lipid droplets, which are storage compartments for triglycerides and cholesterol esters. And they also may serve as platforms for storage of proteins that are targeted for degradation. But in the terms of lipid droplet formation, Cholesterol esters and those triglycerides will accumulate within the hydrophobic core of this lipid bilayer. And then delamination of the two lipid monolayers, so delamination meaning pulling apart here, forms what we like a lens. So you can kind of see that it looks kind of like an eye, the lens of an eye. The lens grows and creates this spherical droplet and releases by scission of this neck here. And the newly formed droplet then is surrounded by a monolayer, which was derived from the cytosolic leaflets of the ER membrane. And it's important to note that this is a monolayer. And why is it a monolayer instead of a bilayer? Well, these excess fats and lipids, those are hydrophobic, right? So the hydrophobic tails of this lipid phospholipid monolayer, they want to interact with those, as opposed to if it was water, they'd want another internal layer so the tails could be together. But since the lipids are nonpolar or hydrophobic and the tails are hydrophobic, they go together very well. So let's take a look at some of these transmembrane proteins. This protein is glycophorin A, which is a typical single-pass transmembrane protein. What we mean by single pass is that this protein has one part of its amino acid chain going across the membrane. Now, you see here a green and a red section going across. Well, that's because glycophorin A is a dimer. So it requires two copies of that protein, each with a single pass, to make up the tertiary structure. So in terms of membrane proteins in general, the kinds and amounts vary depending on cell type as well as the subcellular localization. An example in yeast is that 1/3 of all the genes that encode for proteins in yeast encode for membrane proteins, and this is higher in higher eukaryotes. So there's a lot of different proteins that are in the membrane. So looking at this dimeric glycophorin, as we said, it's a single pass that has only one membrane spanning domain. And this membrane spanning domain is 23 amino acids, and it's in the shape of an alpha helix as its secondary structure. And this alpha helix is composed of amino acids that have hydrophobic uncharged side chains. And why is that important? Well, if you're going to span the inner part of the membrane, the hydrophobic part of the membrane here, you also want to be hydrophobic because you don't want to have any force pushing away on that hydrophobic, those side chains there. And so this is then stabilized by hydrophilic residues where the polar heads will be. So you have hydrophilic wanting to be by these hydrophilic polar head groups, and then hydrophobic in the center. So they kind of mirror that polarity of the phospholipid bilayer. And so the extracellular domain is heavily glycosylated with carbohydrate chains that are attached to specific serine, threonine, and asparagine residues, while the cytosolic domain, that usually interacts with the cytoskeletal proteins, which kind of anchor and allow motility, directed motility as opposed to random floating of this transmembrane protein. And so on the right-hand side here, we have just kind of a 3D structure of this glycophorin A dimer. So our last example was a single-pass protein, but we also have multi-pass membrane proteins. So two examples here are bacterioreactive rhodospins, which is kind of similar to a photoreceptor in the eye. It's actually a photoreceptor in bacteria that allows bacteria to sense light. And as you can see here, we have multiple alpha helices that are spanning this membrane with a retinal protein in the middle here. And I guess that makes sense, right? Because if you are a bacteria and you want to sense light, you probably don't want that retinol in the inside of your cell. You want it exposed to the outside where the light is going to be. We also have an example of a glycerol channel. So this again is a tube made up of these alpha helices, so multiple passes back and forth across the cellular membrane. And this creates a channel for the glycerol to move from the external environment to the cytosol. And as you can see here, this is just a rotation of 180 degrees, and they have the glycerol molecule within the center of the channel here. So we can also have multimeric membrane proteins, and these are proteins made up of multiple different subunits, not necessarily dimers, two copies of the same protein, but different subunits of different proteins to make one macromolecule or macroprotein. So the way that these come together within the lipid bilayer is based on the charge of some of the amino acids within their chain. And so here we have an example of a T cell receptor for antigens, and this is composed of 4 separate dimers. The alpha beta dimer, which is up here, that is directly responsible for the recognition of the antigen, but the CD3 complex here, which is composed of these 3 dimer pairs, they utilize electrostatic attraction of the positive and negative charges on each transmembrane domain to form this complex. And so what you get as a result is this fully assembled TCR, which it was the T cell receptor. And you see you have the alpha beta unit in the top here, but the entire complex that needs to be stabilized is made up of multiple subunits. And as you can see, the pairing of negative and positive charge in this transmembrane area, the hydrophobic area, is what holds this complex together. So in addition to alpha helices, beta sheets can also span the membrane. So here we have an example of a porin, which is found in the outer membrane of E. coli. And so multiple beta strands in porins form this membrane spanning barrel. This barrel-shaped subunit has alternating outward-facing hydrophobic side chains. So these are right here. So those are going to want to interact with these tails that are present in the lipid bilayer. And they also have alternating inward-facing hydrophilic side chains to line the pore. So this is to ensure that there is liquid and not more lipid bilayer chains on the inside of the pore. And so what that does is that means that the hydrophilic nature of the external and the internal environment match what's inside of this pore. And then that will allow water-filled transmembrane pore to allow hydrophilic components to diffuse through. So whether it's like a calcium channel or something that is water soluble to allow to pass from the exterior to the interior of the plasma membrane. So let's look at these antigens on the membrane of blood cells. So there are 3 structurally related oligosaccharide components of certain glycoproteins and glycolipids, and these are on the surface of red blood cells and other cell types. The terminal oligosaccharide sugars distinguish the difference between O, A, B, and AB blood types or antigens. The presence or absence of glycosyltransferases that add galactose or N-acetylgalactosamine or both determine the person's blood type. So let's break this down. So we have our normal lipid or protein here, and if there is no presence of a transferase, we have an O blood type. Now, if you have this GAL-NAC transferase, you are an A blood type because that adds that N-acetylglucosamine. Now, if you have the GAL transferase, you are a B blood type because it adds that galactose to the end of the protein. If you have both, then you're an AB blood type because it adds both to the antigen position on the lipid or protein. So this blood type is very important for blood donation, right, or for transfusions. And so if you are an A blood type, you have A antigens on your red blood cells, and that means that your serum antibodies in your blood are anti-B. And so if you get added B blood, that means that that blood will be destroyed by these antibodies. So you can receive A or O because O has no antigens on the blood type. If you are B, your antigens on the blood cells are B, your serum antibodies are anti-A, and so you can receive only B and O. If you are blood type AB, your antigens are both A and B. There aren't any antibodies because if it attacked A, it would attack its own antibodies. If it attacked B, it would attack its own antibodies. And therefore, you can receive any type of blood. And then if you are O, you have no antigens, and your serum type are anti-A and anti-B, so you can only receive O. So O is the universal blood type for donors, and that is because if you look over here, O can be received by everybody. But people who are O in terms of blood type can only receive O blood type. So we talked a little about those phospholipases, but let's talk about how they interact with the cell membrane. So these phospholipases have lipid binding motifs that target peripheral proteins to the membrane. So these motifs here are these blue sites there. And the enzymatic activity of each of these phospholipases hydrolyzes a specific bond in that phospholipid. And the function of this is to either to degrade damaged and aged cellular membranes, so to take out kind of the broken components, or to generate signaling molecules as well. We have a lot of new insights into lipid signaling and lipid rafts, as they're called. So in the example of phospholipase A2, it has this lipid binding rim of positively charged arginine and lysine residues that surround this catalytic active site. So the active site is there. And the positively charged binding site rim residues bind to the negatively charged polar groups on the membrane surface. So here we have positively charged residues, and then we have negatively charged residues on the surface there, so they bind that way. And when they bind, this creates a small conformational change that opens up that channel and exposes the catalytic site, and this channel is lined with hydrophobic amino acids, and so that will allow this chain, the tail group, to enter. And so the phospholipid moves from the membrane, or moves the membrane leaflet into the channel. And to facilitate this, you would say, okay, well, maybe why is it going into this hydrophobic channel if you have a hydrophilic head group? Well, there's an enzyme-bound calcium ion that binds to that head group. So we can see that right here and in the molecular structure right there. And that allows that polar head group's desire to be bound to a positively charged ion to be fulfilled, and then allows that leaflet to move into the channel, be positioned at the accurate active site, and then be cleaved. So detergents are these amphipathic molecules that disrupt membranes by intercalating, which means kind of penetrating phospholipid bilayers. So sodium deoxycholate is actually a natural salt that is found in bile, but the other examples of these, such as SDS and Triton X, which are commonly used in labs, and octyl glucoside, excuse me, are not naturally occurring. And so we have two different types. We have ionic detergents and then non-ionic detergents. And if you look at these, you notice that they have kind of this similarly colored pattern here. And so there is a hydrophobic region, and that is the region that interacts or intercalates into the membrane by interacting with the lipids. And then there's the hydrophilic regions, which interacts with water to help solubilize the liquids, and so, or the lipids, excuse me. And so what they are doing is they are satisfying those hydrophobic bonds, but then also reacting with water to break apart those micelles or phospholipid bilayers. And so we have non-ionic detergents, and these do not denature proteins, and so they're useful for solubilizing intermembrane or integral membrane proteins. So they'll keep the proteins intact, but they'll put them into solution and break apart the membrane. But then there are also ionic detergents, which disrupts ionic and hydrogen bonds, and it denatures the protein. So it gets rid of that secondary, tertiary, quaternary structure. And so SDS here, if we're doing SDS page and then electrophoresis to look at the size of a protein, it doesn't break up its actual secondary structure. So we get an accurate measure of how big this protein is and its conformation when we're looking to run a gel. But then something like Triton X, that will break apart that protein, all the hydrogen bonds and things like that, and just make it kind of a linear solubilized amino acid chain. So if these bilipid or phospholipid bilayers want to form these layers because they have a hydrophobic side and a hydrophilic side, and they found this way to pair up together to both be happy, well, how do you move single phospholipids through hydrophilic media such as the cytosol, because those tails aren't going to want to be there. And so what there are these things called fatty acid binding proteins. And these are small cytosolic proteins that facilitate the movement of fatty acids. And the way they do this is they contain a hydrophobic pocket that's lined by beta sheets, that is indicated in the yellow here. And this binds to that hydrophobic chain. And then the hydrophilic side can be exposed, or it can interact non-covalently with hydrophobic or with hydrophilic residues near the mouth of the pocket. So let's look at phospholipid synthesis in the ER membrane. So cells synthesize new membranes by expansion of existing membranes. And so membrane-associated enzymes catalyze the steps of this phospholipid synthesis at the interface between a membrane, such as the membrane in the smooth ER here, and the cytosol. So the first step here is that two fatty acids are synthesized on fatty acyl-CoA, and this is esterified to the phosphorylated glycerol backbone, which is right here. And this forms phosphatidic acid. And the hydrocarbon tail of this phosphatidic acid anchors the molecule to the membrane. So here, we have our phosphatidic acid that is anchored to the ER membrane on the cytosol side of the membrane. And so now in step 2, phosphatase converts phosphatidic acid into diacylglycerol, and that is by removing that phosphate group, right, which is leaving here. And so what we're left with is this diacylglycerol. Now in step 3, phosphotransferase transfers a polar head group. Remember, right now all we have is the nonpolar tail that's kind of anchored into this ER membrane, and so we need a polar head group. And so in this case, phosphorylcholine is added from cytosine diphosphocholane. So this is CDP choline here. So that head group is transferred using choline phosphotransferase to attach to that diacylglycerol. And when it does this, we then have our phospholipid, phosphatidylcholine. And so we've created now a phospholipid on the inside of this, or on the cytosolic side, excuse me, of this ER membrane. Now, if we only added to the cytosolic side, we would eventually have this very unbalanced membrane that is getting more and more round, right, and to the point where it's going to burst. So we have to be able to transfer some of these phospholipids from the cytosolic side to the lumen side. And so in some cases then, the phospholipids interact with flippase. And what flippase does is it uses ATP to catalyze the movement of these phospholipids from the cytosolic side to the exoplasmic side, which equalizes the growth and establishes this symmetry across both the cytosolic and the lumen, or the external environment side. So let's talk about how cholesterol is produced. So cholesterol is synthesized by enzymes in the cytosol and the endoplasmic reticulum, and this mainly occurs in liver cells. And so the way this works is HMG-CoA reductase, which is the regulated rate controlling step, converts beta-hydroxy, beta-methylglutaryl-CoA into mevalonate. So mevalonate is this intermediate product that HMG-CoA is turned into through the work of HMG-CoA reductase. So how is this regulated? Well, when there are high levels of cholesterol in the ER membrane, cholesterol will bind to that HMG-CoA reductase, to this, and through interactions with other integral ER membrane proteins, it will induce the role of ubiquitin to come in and tag those HMG-CoA reductases for degradation and reduce the amount of those HMG-CoA reductases. So it reduces the capability to form more cholesterol. And so once these are tagged with those ubiquitin, these are very bad drawings, ubiquitin tags, it'll send that to the proteome, which breaks it down into little pieces, right? And so that prevents more cholesterol from being synthesized. And that's all based on the mevalonate intermediate. So in unfortunate people like me who have high cholesterol and they take statin drugs that are used to treat high cholesterol inhibit HMG-CoA reductase, which then decreases your natural cholesterol synthesis. So it lowers the amount of cholesterol that you have in your bloodstream. And so mevalonate is converted into isopentenyl pyrophosphate, or IPP, which is a basic 5-carbon isoprenoid structure. IPP is converted into cholesterol and other lipids, often through the polyisoprenoid intermediates. And so, keep moving down here. And these intermediates are then turned into either cholesterol or numerous other compounds, which include lipid anchors that will anchor proteins to things like the actin cytoskeleton or the cellular membrane. So how do cholesterol and phospholipids transfer between membranes? So if they're created in the ER membrane, how do they get to the cellular membrane or to the mitochondrial membrane or the nuclear membrane, or all these different membranes that occur within the cell? And so there are 3 proposed mechanisms for how this works. One, the first mechanism here, is that there's this budding kind of endocytosis type movement between these membranes, where a vesicle buds off the membrane and fuses with the target membrane to transfer lipids and cholesterol between the two. So we have this movement like that. The second is that lipids are transferred directly by membrane-enabled proteins by contacting these 2 membranes together. And so that would be that these membranes kind of squeeze together in these 2 membranes on the opposite, or proteins on the opposite membranes here, kind of act as magnets to pull those membranes together, and that'll allow the movement of leaflets or proteins or cholesterol to move from one membrane to the other. And the last is this idea that there might be these small soluble lipid transfer proteins that in the case of, that would be specific for cholesterol, and another one specific for these phospholipids that could transfer from one to the other. So that wraps up this chapter. I will look for the quiz for chapter 7, and I will see you in chapter 8.